there was something. All right. So last time, I think we sort of ended up writing something quickly on the board that looked like how this does not. Oh, all right. How a population of two types evolve, and if you have two subtypes, we could write down an equation that captures the effect of selection, mutation, and also stochasticity by binomial sampling in the population. And then we characterize the noise, and we also sort of projected this to a frequency, because basically that's the only independent coordinate in the system that matters. Some of the subpopulations, so the total population size is constant, so the only thing we care about in this case would be the frequency of, let's say, the green guys. And so this was the thing we wrote down. And so this is basically the simplest toy model of evolution that you can put together, right? So two alleles, single locus. So I'm trying to sort of use the normal population genetics jargon here. So locus or loci are, you can think about the places in the genome that encode for some function you care about. It doesn't have to be a nucleotide, and we're going to see there are versions of that are not nucleotides. And then the alleles here, so these are the types that are present in principle in a population. You can think about this as different amino acids, or different sort of, if you have a population of wild type versus some mutant, and these are the only things that are present, so one you can call it the up arrow and the other one the down arrow. So it's just a matter of what you care about. But yeah, so degrees of freedom in your system, so to speak. All right, so to remind you a bit of sort of a stochastic process notion, I'm going to go with this a bit quickly maybe. How many of you know basics of stochastic processes? Okay, then I can go quickly. Good. All right, so the thing we wrote down is a Langevin equation, right? So it's basically, if you're a mathematician, you would hate this form. But as physicists, we still write it. And there is sort of a rigorous way of going from this equation to what is known as a Fokker-Planck equation, which would characterize the probability density of now your random variable x in ensembles of populations. So the Langevin equation characterizes one trajectory, the dynamics of one trajectory. You can write a code, and you basically simulate this process, and at each step you add some noise with the statistics, and you sort of look at the dynamics. Now you make ensembles of these trajectories, and this is the probability density of the state. You could look at probability densities of the trajectories, and we're going to get to that later, hopefully today. But right, so you can go from here to here. This probability density is in principle, time dependent and state dependent. If you set the left-hand side of this equation to 0, then you end up characterizing the stationary state of this. The stationary state and equilibrium are not equivalent necessarily. So if your current is constant, you can have a stationary state, but if the current is absolutely 0, then you have equilibrium. But in the case of probability density, because it is conserved, and because we're looking at a one-dimensional case, so there's no curl in the system, if you characterize the equilibrium process, you're actually characterize a stationary state, you actually characterize equilibrium. Is that clear? All right, there's no objection. So in this case, you can actually solve for the equilibrium. So let's assume for the moment that your selection is 0. So we call that neutral evolution. So you have only the component of noise, so the genetic drift stochasticity noise and the sort of deterministic part from mutations. Can anyone guess what at least the mean of the x would be in the stationary state without any selection? So what's the average frequency you would get just by fluctuation and mutations? 1/2, right? So it's basically completely unbiased. Now you tune up selection one way or the other, you sort of bias your system, having more of one type or less of that type, right? And but the distribution also has some interesting features, right? So if x is equal to 0 or equal to 1, you get basically no stochastic component, right? So this is what is known as a multiplicative noise, right? Depends on the state. And right, so the sort of the drift term, the normal drift term, right? So the deterministic term vanishes at x equal 1/2, so that's your fixed point, and the deterministic state. All right, so if you characterize the equilibrium distribution in the neutral regime, so for now no selection, you end up having this probability density function. You expect it to be symmetric, right? So you don't care whether your x is 0 or 1. So it should be symmetric around 1/2, and that's what it is. Okay, so this is one case of this. So mu n is a parameter, right? So this is a probability density as a function of x. I should have put the axis. So if you put mu n smaller than 1/2, you get the shape that looks like this. So it's called U-shape Kimura distribution. And because this equation is diffusion, Kimura's diffusion approximation, and Kimura was a person who characterizes these things in 1960s. So if you increase mu n, so mu is a mutation rate, right? So if the mutation rate is very small, that means that you're spending most of your time at the boundaries, right? So if you think about the sort of the trajectories we were plotting before, so if your mutation rate is very small, you're either in one boundary or the other. Once in a while, you do the jumps. If your mutation rate is very high, what happens is that you're sort of spending most of your time in the middle, right? So can anyone have a guess what will happen if I sort of increase mu n and go far above 1/2? So what would be the limit of this distribution? So the shape of this distribution at the sort of large mu n regime. Yeah? Yeah, it'll look like a Gaussian around 1/2, right? Because basically you're not anymore spending most of your time at the boundaries, and these are the sort of the singularities you see. You spend most of your time in the middle, and it sort of looks like a Gaussian, as things tend to look like a Gaussian. But regardless of that, you can also characterize the sort of statistics of this distribution, and I just wrote down two. The first one you guessed. The second one is this quantity, and I think we mentioned that x1 minus x would be the variance of your states in a population, right? So the average, ensemble average of this variance is equal to the mutation rate times the population size. And that's the thing you can sort of guess. So it's the amount of segregating alleles in a population. So the more mutations you have, the more variations you get in a population. And that's sort of intuitive that you would get this sort of quantity as your ensemble average variance. It has some significant sort of practical outcome, right? So if I tell you, so n is sort of it's not a true population size, so it's not a human population size, it's what's known as effective population size. And basically it's a quantity that tunes the amount of noise in your system. And for humans is approximated to be 1,000 rather than 10 to the 8, right? So the way you sort of try to guess n is you try to fit more or less a dynamics like this to what you see in your genomic observables. And one easy way of doing this is you find places in your genome that look pretty neutral, and you characterize how many, and then you go sequence people or flies or bacteria or whatever you care about, and then you count how much variation you have at individual sites. And so mutation rate is a much easier thing to characterize. You can measure it in the lab, for example, in bacteria. You see how many mutations you get per generation. But what you end up getting is that you can also characterize the variance, and so you get the mu n out of that, and you can estimate the effective population size. The other way of doing that is a process of coalescence. Which is the more proper way of doing that. So if you have a population that sort of is randomly mating, and you try to sort of find common ancestors of the sequences that you observe, right? So this is what you observe. You try to reconstruct the past by looking at commonality of their genomes or connecting them to their parents. And let's say the mating is random. So what's the probability that, let's say, two guys I pick have the same parent if this is population of size n? So what's the probability that these two join to the same parent? It's 1 over n, right? So it's just I randomly, so this guy has one parent, and I randomly pick a parent, and the probability that I hit the same parent is 1 over n, right? So then you can sort of translate that to rates and time. So the sort of the expected coalescence time for individuals in this setting would be n. So population size, in a sense, is some measure of time, if you wish, which is not intuitive because it's just numbers, but it's a measure of coalescence time. So that is another way of looking at sort of trying to infer the coalescence time in the neutral regime and inferring n. All right, so all this said is an effective parameter. It has more or less nothing to do with real population size. It has something to do with bottlenecks. So the population size that mate together and are together, and you sort of they experience the same as stochasticity. All right. So now I turn on selection of this form, and I can still evaluate the probability density. So this was what we had before, and I call it P0 of x because this is a neutral probability density, right? So that's the 0. And then you end up getting sort of a factor e to the 2n, this sort of the difference between their fitness times x. And you can show that up to a constant, that value is actually the mean fitness of a population. So if I were to calculate the mean fitness, it's basically x times fa plus 1 minus x times fb, which is x times fa minus fb plus the fb, which is a constant, so it doesn't matter. You can absorb that in a sort of in your normalization factor, and this is your delta fab. So I can rewrite that sort of red thing as 2n times the average f up to a constant, and constants don't matter. And this is a consequence of the structure of this Fokker-Planck equation that we have here. And we're going to get back to that even in a more involved way. But just if you look at this, right, so you have a variance here that appears in front of your sort of second derivative. The same term appears in front of your fitness difference. So now you can think of this as if your fitness is a derivative of some potential function, if my delta fab is a derivative of some, it's called v potential function, right? Then what I end up having is I can write down the sort of stochastic part and the selection part together as 1 over, well, there's some the derivative, second derivative with respect to x, 1 over 2n x 1 minus x, right? And then minus second derivative times x1 minus x, right? And so this is sort of accounting for a system living in a potential and looking at the stochastic process in a potential, right? And the solution of these sort of processes will always look like that, right? So you have a sort of multiplicative noise term, and it can be as complex as you wish. But if the same covariance coupled, the same fluctuation coupled to your sort of potential function, then you end up having a solution that looks like, or equilibrium solution that looks like a constant time e to the potential, right? So e to your Hamiltonian, e to your energy, right? All these things. And the potential in this case, you can easily guess what it should be. It should be basically the mean fitness in a population, right? Which is delta FAB times x up to a constant. So this term is your potential fitness potential, and you're basically writing down something that looks like a Boltzmann distribution. Is this somewhat clear to everyone? Right. All right. And then you can build up your analogies, right? So then n is playing the role of inverse temperature in this case. So basically you're tuning your fluctuations like temperature would tune fluctuations in a thermal system. And then you also have an energy term that is the mean fitness. But there is a sort of significant difference here, right? In thermodynamics or in physical systems, what we deal with is we have a conservation of energy normally, right? So we have second law of thermodynamics. There's nothing called conservation of fitness here because we discussed that there's sort of this gauge invariance, right? So everything, just the relative fitness matters. The absolute fitness doesn't matter in what we are discussing. So you can sort of shift everything up to a constant and you get the same form. So, all right, is this somewhat clear? So I'm sort of going into unnecessary analogy and details, but it'll become more clear as we build up the formalism, how to use these ideas to now sort of go and look at the more complicated problems. Yes?
So I just wanted to clarify, so n is like an effective population size based on content nature.
Random mating.
Right, because so you're basically saying that you could have like a much larger population, but if they're all kind of only interacting
Locally, yeah. Then it doesn't matter, yeah. Yeah, exactly. Yeah. All right. Okay, yeah?
Curious, what do we have any evidence that the n has increased as the airplanes have developed or something along those lines?
I don't know. Good question.
Population size increase?
Yeah.
Like for us? Evolutionary time scale.
Yeah,
The
so not
airplanes,
evidence-
don't count on evolutionary time scale.
Yeah, but I mean, the population size has gone through multiple bottlenecks. That much I know, but apart from that,
No, but look.
but that's an
I think
evolutionary
apart
time scale.
from aeroplanes, the other thing that's happened is the exponential populations, I suppose. So, I mean,
Yeah, so that's another thing.
there's things that have happened in the last 100 years that I think are more important than airplanes. And if you think historically, I mean, think of Genghis Khan. I mean, he traveled as far as you do by airplane.
Yeah, but okay, so the exponential increase in population size doesn't mean necessarily exponential increase in effective populations.
No, sure, But
So if everyone lives in their own village still, no one cares,
yes, but
right?
people traveled. I mean, we have this misconception that they lived in the little villages, but they traveled everywhere, right? I mean, okay, now we do it.
Not everyone.
But not everyone propagated everywhere.
And
So
still
maybe
not
Genghis
everyone-
Khan did,
and
but
still not everyone is on the right plane everywhere. You know, I mean, and anyway, I mean, I
And
think
also some people travel by plane not to mate. That happens too.
What are we doing?
Because I think that was your question, yeah? I know what you come
are we going into more details now, or what's
that was the
place?
question, Ilya.
I just want to understand where this number of a few thousand comes from.
I think, okay,
And
so
is it changing as it's changing, but yes.
it's changing, but I think
I
it's
mean-
not the plane that is responsible.
Yeah.
But the airplanes are responsible for mixing of pathogens. I think that's a more interesting sort of
And
consequence.
climate change.
Yeah.
And we shouldn't really be here, so let's keep that plane slow.
A note to the organizers, no? Okay, so you want to make a joke, maybe airplane joke?
I do have an airplane joke.
All right. So this was a super sort of simple toy model. Is it good for anything? And the answer is, you can always find simple things that your simple ideas might fit to. But in this case, there are, so I sort of use the term locus and allele, not nucleotide and genomic position and sort of this kind of things to have a general sort of scheme of where the function is encoded, right? And what we care about. So we looked at these numbers the first day and also partly yesterday. So how much circulating sort of variance you have in different populations. And this is the example of bunch of populations. So you can look at the mammals. So this is the sort of genomic variation in some mammals starting from monkey or baboon to hyena and foxes and things like that. So you get basically from 10 to the -3 to 10 to the -4, something around that range. So basically 1 in every 1,000 nucleotide differences or every 10,000, yes?
Is new mutation rate per individual?
Per nucleotide per generation, right? So it's not within an individual, right? So mu n was this variance of loci, right? So that's this average x1 minus x. And that's why it's actually observable, right? So n on its own is not observable, yes?
So, all of the species in this graph are sexually reproducing. Shouldn't that change a lot of things?
Yeah, it should, but we don't get into that. It's a factor 2 or 4 or whatever you care about, right? So and there's recombination. So let's keep it simple. I want to just tell you basically what the range of these numbers are, right? So the butterflies live in 1% regime, at least these guys, right? Drosophila also lives in 1% regime. So these are the things you can get the data from easily, right? So we sequence them. We have what's known as polymorphism data. So that means you have within population variance of that, and we don't have that polymorphism for everyone, right? So what that implies is that if I find a region in the genome that is 10 base pair long, for example, and I look at the population and count how many variants, genomic variants I have within a population, if this is the amount of variation I expect, more or less the whole population should carry the same allele, right? In that region of 10 base pair. It's not a single nucleotide, but it's a short enough region that you don't expect to see much variation in a population. Just, multiplying 10 to the -4 to 10, yeah.
Do you know what the color code is in that bar?
Different animals. Oh, no. No, yeah, I don't know. Sorry. Well, these are 2 humans. No, I can't come up with this story, sorry. All right, so we heard about these numbers yesterday. So gene length in humans or eukaryotes, so order of kb, kilobase per. Regulatory genomes, depending what exactly you define as regulatory genome, it can go from 100 to 1,000 base pairs. So this is like many binding sites, promoters, enhancer you put together, so as functional regions. You can then also look at the smaller units, a transcription factor binding site. So that's the place that the transcription factor would bind on the DNA. So that's a much shorter region, so something of order of tens of nucleotides. Again, depending on the species you're looking at, but this is now the unit that is an interesting unit and our toy model might apply to, right? So it's short enough that you don't see huge variation within a population there. So you're either having maybe one allele or the other present. And see how far we can go if we look at the sort of transcription factor binding site evolution. All right. So this is our transcription factor. So let's say it binds to a region, sort of sequence A, A1 to AK, so K base pair of order 10. And then let's say, so the thing that matters, and I sort of gonna emphasize on this term, which is not bold-faced for some reason, molecular phenotype. So the thing that matters for the transcription factor binding site, right, that defines its fitness is how well it binds to the transcription factor, right? So there's a quantity you can sort of characterize and you can measure it. There's binding energy that matters, or affinity of this interaction. And we will call this sort of molecular sort of features as molecular phenotypes. So this is not the height of an individual, it's rather something in a much sort of smaller scale, is at the molecular level. And we have now access to data measuring binding energy between transcription factors and sort of the DNA. That's one molecular phenotype. The amount of the amount of activity of a gene, so the gene expression level, that can be another molecular phenotype, slightly more complex than this one. So I'm going to talk about these molecular phenotypes more or less from now on, and this is the simplest one to start with, basically. All right, so this defines the fitness of this binding site, and you can now go in the genome. So let's say you have a model of a sequence to binding energy map, right? So I give you, and that's a big if, right? So it's not necessarily an easy thing to infer, but let's say someone give you a matrix that you give a sequence. Let's say it's even a linear model, right? So you go to sequence, you just look at the matrix, at what position, what nucleotide is there, and then you sort of give me a binding energy, right? And so you can then go and look at the whole genome. And let's say you know where the real binding sites are, the functional binding sites, and where the sort of random pieces of the rest of the genome, basically, that are not the binding sites. And this is what happens. So this is the specific binding site or transcription factor in yeast. And this is the distribution of these binding energies. So the Q of E is the distribution of these binding energies in the among the functional sites. So you collect your functional sites, you measure the binding energy, and you look at the distribution. So small e is good, so we're still in the physics realm, so high affinity. And then the other one, so the blue line and the dots are the so the blue line is the sort of the null distribution, so to speak, so non-functional site. And what you see, actually, the dots here is when you put everything together in the genome and you don't know what's functional and what's not, so the dots are actually the data. And you can then try to decompose the data into these two distributions and call the sort of this little one as a functional distribution and the blue line as a neutral distribution. Right. And so you can sort of think of this now as two states. there's a functional state with high selection, and there's sort of a neutral state with low selection coefficient. And our theory of sort of two allele single locus, if you compare across species, might hold for individual sites here. And if you were to try to relate this distribution to what we just learned in population genetics, what end up being the case is we know what the fitness is. Now, let's say we have an energy-dependent fitness. The fitness depends on energy of each site. We know the mutations. So mutations are few. They're rare. Maybe you just change one site to another. And then what you can also calculate is, if this is the model of evolution, you can calculate from your Kimura equation that we wrote, you can calculate what's the probability of fixing a specific allele by a certain time in your population. So you start from it starts from one individual with that type, let's say the upward arrow, and you ask what's the probability that everyone else in the population will be upward arrow just by mutation and selection. And for that you need to calculate the backward Kolmogorov equation. So we wrote down the forward one. Is everyone familiar with this? All right, good. And so if you calculate that, this is basically give you this formula that relates what is known as substitution rate. So the probability to get a frequency equal to 1, starting from basically one individual. The guy substitutes. So these were the big jumps in our plot that we had, right? So we had sort of x equals 0, x equals 1. So we had this sort of wiggles, and then once in a while you get this, right? Then you get wiggles, and then you go down. So these are the substitution events, right? The big jumps. And you can ask what's the rate of substitution from one type to another, and that you can calculate. And I'd be happy to discuss with you how to calculate this. But to get an intuition of this, if your fitness is large and positive, you end up basically going a lot from B to A. Yes?
For a long time?
Yeah. It's a sort of hitting probability, right? So, right, so if your fitness of A is larger than B, so delta FAB is positive, right? Then you tend to transition a lot from B to A. And so in the limit of delta FAB being large, this thing becomes this formula becomes linear. So and it'll be proportional to the fitness difference. If delta FAB is is negative, so A is actually deleterious, you're exponentially suppressing that allele in your population. And that's basically this exponential negative tail on this side. So we can characterize with what rate you switch from one state to another state, right? We are in equilibrium, and we have detailed balance. So that means we actually know what's the relative sort of abundance of these two states in a population, right? So we have a formula like this. So the probability of being in a state A over a state B is inversely proportional to the rate going from B to A divided by A to B, right? So that's the detailed balance. And we know what exactly that is. And now if you go back to your data sort of projection and define the neutral state versus functional state as the two states you have in a population, and trying to characterize with this formula, sort of interpret this as some evolutionary fixation, right? So you're sort of having these two distributions, and you're trying to basically assign a fitness difference that would put some of your sites into a low energy or high affinity regime and make them functional. And okay, does anyone have a guess how to actually characterize fitness in this case? So what we have here is, let's say you call P0 of A or P, so Q of A, put it in the numerator, you put P0 of A in the denominator, right? And then let's say mutation rate is the same forward and backward, so this is a neutral mutation rate. What we get is that the states will, so what the selected state would have an exponential weighing up there, right? So which would be proportional to its fitness. And what you end up getting is the sort of the relative probability density would be equal to e to the 2n fitness, or the log likelihood, or the sort of log likelihood ratio between the two would be equal to the fitness of the functional one. This is a thing that's used in the statistics a lot, right? So you sort of characterize the likelihood function, and you have a null model, and you assign fitness to it. But then you can have actually an evolutionary interpretation of this likelihood function. And you can then go, and from data, you can try to characterize basically what the fitness landscape should be as a function of energy, right? So we know the functional size, they're all in the small energy regime, so that means they have the high fitness. So this ratio is larger here, and then at some point it drops because we don't see any functional site here anymore, right? So at some point it drops, and basically your fitness becomes very small and saturates, yeah?
I'm a bit confused. Is that you have two states, that is
The neutral,
and then the selectional one, and
Yep.
then each of them have many energy. So how are the energy and the two states related to each other?
right? So basically you sort of think of every site that you see as one draw from one distribution or the other, right? So that's how these energies are, right? So you can think, okay, I have a site here. On evolutionary time, the site can switch between being functional or dysfunctional, right, in a population. But then I have many sort of examples of that in the genome, and that gives me the distributions of the energy. So these are sort of ensemble characteristics, and then the transition happens for each site, right? So we are mixing the ensembles together, right? So we think that, okay, so these are all examples that are drawn from the same distribution. And that's where the energies come from. Yeah?
I mean, just to clarify for myself, so basically the idea is that you observe frequencies of different mutations,
Mm-hmm.
and then from there, and you know, if you knew the fitness, you would know that the relative
Yeah.
rates of transitions would depend on the fitness.
Yeah.
And then you say, so at the same time, you can compute energy as a function of
Sequence.
So the question, so what you're trying to do is to infer fitness as a function of energy.
Mm-hmm.
What we have on the right. So what is this hidden fitness
Yeah.
that in sequences I only under,
Yeah.
such that we observe what we observe? So how is this fitness related to the energy? But make an assumption that the energy is really the way we compute the energy.
Yeah, I mean, let's say I give you a sort of a recipe to compute the energy, right? That's itself a sort of a question worth discussing, right? And so my observations here are just the states of the system, right? So the fraction of sites with different energy values. And I try to sort of split them between functional and dysfunctional in a way that is consistent with the picture of fitness. And that's what comes out of it. And this all work because we are in the regime that detail balance hold for sort of low mutation or weak mutation in the genome or in the regions we care about. And these are the binding sites, and because they're short, the mu and L is smaller than 1, much smaller than 1, so you don't expect to see so many sort of variants present in a population at a given time. In reality, life is more interesting than individual binding sites. You heard about, sort of complex promoters and enhancers and things interact with each other. And at the end of the day, these are the things that define the function of a sort of a real molecular phenotype, right? So how much a gene is active doesn't depend on individual binding site, but rather on combination of sites and many transcription factors interacting with many sites, nucleosomes here, and so many other things, right? So we are not in the regime of mu and L much smaller than 1 normally, but when we are thinking about things that we care about. you can also think about protein-protein interactions. So that's another phenotype of energies. And so that's also, you have a lot of contact size, and it's much larger than just 10 base pair. So all the sort of things I mentioned so far has been to build up this slide that everything we talked about may be useless in some sense. And so we need to actually move forward, and so go a step forward and try to understand, so how can we formulate evolution in this high-dimensional space in a way that is tractable? Because you can sort of write down a very complex many-body interactions of all possible mutants in a population and all the transitions. I mentioned that you can characterize a stationary state. It doesn't mean that it's equilibrium. And especially if you have many variants, you get basically probability current that have curls, and it's very hard to track these things. And can we do a proper coarse graining to actually address this problem? And that's the thing that we're going to do basically from tomorrow, from the rest of the time. So what I would like to do is to start from what we built so far, and right. And give you first maybe a rigorous description of how the dynamics should be. In a high-dimensional case, how can we interpret the sort of the formula that we write down, and then how can we do a projection to sort of more coarse-grained description of the system. So, all right, so far what we had was two alleles that effectively become 1 degree of freedom, which was just the frequency. So this next step would be having 3 alleles and 2 degrees of freedom, right? So, and I'm not going to drive every details of this because you can just easily do it the way we did it before. So let's say you have A, B, C. You have basically effectively 2 degrees of freedom. The sum of the populations is equal to n. And we can do the same trick, right? You have the noise in n so this is the n noise. Who's basically delta AB, delta t minus t prime, and A. So this is exact same formalism. But now if you were to project this into the frequency space, you get a 2x2 covariance matrix, right, for the 2 independent frequencies. And you can guess that there should be a difference between diagonal terms and off-diagonal terms. Yes?
Sorry, so in this case now we have, I don't know, like instead of now up and down, we have up and down and in the middle something.
And 45 degrees, yeah, exactly.
And so this is still single locus?
This is still single locus, exactly, right. So you can also think, okay, these are 3 genomic variants, right? So if you have a genome of length L, and each of them has 2 states, right? So you have basically 2 to the L possible genomic variant. And so this is the transition from one type to another. So these are just types from now on, right? So if you do the sort of transformation that we discussed last time, right, going from the n noise to the frequency noise, right? So we said chi of xa, right, would be basically the derivative of chi of n, right, sum over I. And what you end up having is this interesting covariance matrix. So if you're on the diagonal side, I of x, A of t, well, let me just write down the two forms. So if A is equal to B, you recover what we had before, which was just x1 minus x. Now you have to write which x down minus xa. And if A is not equal to B, so the off-diagonal terms, you get basically this form. And this is a sort of, okay, so this is a covariance matrix. Now you can expand this, you can formulate it for arbitrary dimensions, k dimensions, right? The same formalism works, the same form of the covariance matrix holds, so so I have diagonal terms would be x1 minus x, and the off-diagonal terms would be minus xi xj. And now sort of trying to relate covariance to metric on this space, so you can now think about this covariance matrix as an inverse of a specific metric on a manifold of in a frequency space, right? In principle, this is a curved manifold, and the reason for that is that we have a constraint on, the sum of your frequencies. It's a trivial curvature that comes from the fact that frequencies sum up to 1. So there is actually a correlation between them. So there's nothing super interesting maybe about it, but it makes life very complicated when we want to write things down. And so in a sense, if you have a metric, let's call it G, G of x, well, let's call this guy G of x, and then the metric would be G alpha beta, right? So this is your covariance matrix, and let's be consistent notation-wise. So this is your covariance matrix, and then you can use the sort of the Einstein notation and define a metric that that is inverse of this covariance matrix, right? So G minus 1 alpha beta, right? And that defines basically distances on this manifold of the frequencies for you, right? So if I move in this sort of manifold, I basically get G alpha beta dx alpha dx beta. So things are not sort of Euclidean additive distances. You have to go.
So this is metrics inversion, right? This is metrics inversion.
Yeah. Yeah. You have to sort of navigate your way in the curved manifold due to the sort of conservation of frequencies, so that they sum up to 1. So this is sort of one formulation of this. So we characterize the noise statistics. Now, we had this formula for how to write a Fokker-Planck equation for frequencies in one dimension. Now you can imagine you can write down for much higher dimensions, arbitrary dimensions. We're not going to drive every single term, but we are sort of using the analogy that we have built to guess what the terms should be. Right. So now instead of a single frequency, I have a vector of frequencies. I have a population size n. So this would be my sort of stochastic part, and the thing that comes up here would be G alpha beta, which is your covariance matrix, which depends on X. Then you have a term that that should be your sort of selection term, right? And what we wrote down before was the first derivative, and then it was the same covariance matrix times some selection coefficient, which was delta FAB. So let's just write down the whole thing. I call that selection coefficient S beta. And then let's say I have a way of actually doing the same thing for mutations. So I can characterize a mutation field as well that couples to my covariance matrix and gives me a field, sort of force here. So if you remember, these were basically the forces that you were getting out of the dynamics, right? And there were the derivatives of your fitness potential, right? In this case, S beta would be the derivative of the mean fitness in your population, and M beta, which is the mutation field, would be the derivative of something that is the mutation potential, right? We never characterize this, and we can try to look at it and what it would be in a one-dimensional case. But what it gives you basically is some sort of generalized, and then there's all sums of alpha, beta, right? So I didn't put the sums. Generalized formulation for high-dimensional allele frequencies in a population. So if you were to guess what M is for one-dimensional case, right? So we knew that the little m is, so G alpha beta was x1 minus x, right? And the little m, so m times G alpha beta was 1 minus 2x times mu, right? So if you try to associate a sort of a potential to this field, there will be some logarithm of x minus some logarithm of 1 minus x, yes?
So fitness doesn't have a curl?
In this case, we assume it has a fitness potential. It could have a curl. It'll be a bit complicated, yes? But even without a curl, even without a fitness, right, so you can produce probability currents with curls in this high-dimensional space. And that means that you don't necessarily have an equilibrium solution for this, right? And so even if you look at the stationary state, it depends on the memory of your system, right? And that's not a good thing. So is there a way to avoid all this and then try to understand things that we care about, right? All right. So let's try to build a sort of simple molecular phenotype version. So evolution of phenotypes in the regime that mu and L is larger than 1, or even of the order of 1. And we want to characterize how things evolve on a fitness landscape, and how do they climb up the fitness peaks, right? And so, all right, to give you sort of a now overview of what we so far talked about, right? So in the sort of genotypic space, in principle, we are dealing with sort of microscopic fluctuations. We are living in a high-dimensional space. So if your sequences are two states instead of ACGT, let's say you have A and C, and then you have L loci, you have 2 to the L possible sequences, so a lot of variance, so high-dimensional. And normally when you characterize fitness as a function of sequence, it's a rugged fitness landscape, meaning that it's not easy to go necessarily from one sequence to a nearby sequence. You can try to criticize these topographic maps, and I'm all for it, but just to give you a sense, there's no smooth transition from one sequence to its nearby sequence necessarily in the fitness world. So dynamics may be rather complicated in this high-dimensional space. Now, there are versions of sort of fitness to genotype maps that have been mapped out. In this case, this is a fitness landscape of a fungus. And looking at a 5-dimensional, or five loci that characterize something relevant for this fungus. And so they measured all the fitness of all the sort of wild type versus mutant variants. And you can already see that the hypercube here may be pretty complicated to navigate on. But, this was already doing a lot of combinatorics. So you can imagine it, how difficult can it be. The other version would be, let's try to do some sort of projection from genotype space to a phenotype space. Let's say we discuss about binding energy as a phenotype, but you can think about gene expression level as a phenotype, protein-protein interaction as a phenotype, and you can also characterize fitness as a function of the phenotype. But then you need to know how evolution projects into the phenotype space to then characterize the dynamics. And that's what we try to do. So the good thing about it is if you do these measurements in the lab, they're normally much smoother than what you would see in a genotype space. They're lower dimensional, but then you end up having some long-range genomic interactions that you produce by doing this projection and some correlations that you need to take care of in a non-trivial way. So if you were to build up the simplest phenotype model, and that's what we're going to write down, let's try to write down a version that is sort of Ising type projection and even a linear phenotype. Okay, so let's say your spins have, instead of 1 minus 1, they have 0 and 1, right? And then you define an energy that is sort of this sum of the spins coupled to some quant variable EI, so different positions have different importance in this setting, right? And we want to characterize how this phenotype, this energy phenotype, changes over time. So if you look at the population, these phenotypes are distributed, right? You don't have only 2 states. You most likely have something that looks like a Gaussian distribution because you have a lot of loci and a lot of variance. To a certain approximation, it's Gaussian. And so you can then just think, okay, can I characterize the evolution of moments of this distribution instead of looking at how the whole probability density changes? If it's Gaussian, you can truncate this at the second moment. If it's more complicated, maybe you have to go to higher moments. Life becomes complicated. So the whole formalism works as, okay, I characterize intra-population phenotype statistics, and I look at the evolution of these statistics over time, and look at ensembles of population and how these statistics vary across ensembles of population. It was the exact same thing we did before. It was the frequency of a certain allele, and we had the probability density of the frequency across ensembles of population, right? How many of you are following? Okay, more than 0. That's good. Is there any question that we need to address before we move forward with this? Yeah?
Would it also make sense to do this for, say, the 2 allele case with 2 sites, with at least 8 states?
It would make sense. You can write it down. I mean, you can write it down, right? So now what you get at, so this is a good thing because you could sort of rigorously write it down, right? Then you can sort of have, maybe small epistatic parameters, then you do perturbations to solve them, right?
But this does not have f-stasis built into it, right?
No, but your fitness can be anything, right? So this quantity can be anything.
But this is one side with many of these.
This is sort of having, in principle, k different genotypes, right? You can have as complex as you want for your mutation sort of field, right? So that defines any transition you want. You can also have as complex as you want a fitness landscape, right? It doesn't mean you can solve it, but you can write it down. It's always a good thing, yeah.
Regarding the point earlier of the fitness being curl-free, wouldn't it make sense because otherwise you would be able to come back to the original position? It wouldn't be conservative.
It wouldn't be conservative, but
which is contrary to. Is there any reason?
there is no reason to have a conserved fitness, right? Because what matters is always a relative fitness at a given time in a population, right?
Okay, so there is time.
Yeah. So. Good. All right. So basically that's what we're going to do. We will sort of try to use this formalism that we had for high-dimensional genotypes and then project it rigorously to the phenotype distributions and characterize the evolution of the moments of these phenotypes. All right, so I think I'll keep that one. It's this thing, right? Maybe I can actually go to this right on the other side. All right, so this is our phenotype. Our sigmas are zeros and ones, right? If you were to write the mean phenotype, which we define as gamma, which is the average E within a population, right? So this would be E I alpha sum over I, and alpha basically are the types in a population, so genotypic types. And then you have, sorry, I can do it. So E alpha X alpha, so the energy of a type alpha times the frequency of the type alpha. So this can be, if you have a genome of length L, 4 to the L types in a population. And so if you think that, we are in the continuous regime and Gaussian approximation is a good thing, then you can approximate this by integral, right? That would be just E, let's call the distribution of energies in a population W. So it would be E W of E dE. So that's basically the mean phenotype of your population. Then we can have the variance of phenotype. And with the same thing would be basically E minus gamma square W of E. Right. So we're going to look at this sort of super simple fitness landscape. It has a name. It's called the stabilizing fitness landscape, and the reason for that is that if your gray thing is your population, if the gray is your population and you have a fitness landscape with a single peak, that means that the population stabilizes around that peak. And so you have an optimal phenotypic value that you care about. And you may think that might be a reasonable approximation for a lot of phenotypes you have, at least locally around some region, you might have an optimum, right, that you want to be close to. And so if you turn on selection for this, you end up pushing the mean of the phenotype towards this sort of optimal value, and also reducing the variance of the phenotype that normally is pretty broad. If you turn on selection, you're sort of restricting individuals to all be close to the peak. So the fitness, in this case F, as a function of the energy, would be basically minus some constant times E minus E star squared. So a quadratic fitness landscape. All right. So if I were to write what's the average fitness in a population, and we write the average fitness because we know how to then do derivatives to get to couple them to covariance matrix and solve things. So the average fitness in a population would be basically the integral of f of e, w of e dE. And that's a simple Gaussian integral, right? So and then the solution would be just minus C0 gamma minus E star squared minus C0 times delta, right? So there is a component that comes from your mean phenotype, and there's a component that comes from the variance of the phenotype in your population, and they add to the mean fitness of a population. So the fitness of individuals should all be sort of compared to this mean fitness. If you're higher than the average, you grow faster, and if you're lower than the average, you grow slower, right? And now that we have this, so we want to know how different how mutations and selection and genetic drift basically impact the changes of this phenotype statistics. All right, so let's do selection on the mean. All right. So does anyone have a suggestion how to go to the next step here? So what is actually changing on the selection? It's the sort of frequency of types, right? Or the frequency of now, we only care about types in the context of energy, right? So the frequency of different energies in a population, or the sort of the W, right? So now if you're changing the W, it depends, the way you change it, it depends on its fitness, right? Relative to the mean fitness in a population. So any suggestion how I move the derivative with respect to time inside the integral? So this is in principle time dependent thing. So you can think how we changed the frequencies when we wrote down the one-dimensional thing, right? So how the dx dt was changing, right? It was some sort of trying to see how the fitness compares to the mean fitness of the population, right, times x. So it goes down exactly the same way. So the W of E would be basically changing the probability density at state E would be fitness of E minus the sort of mean fitness in a population, the capital F, W of E dE. Right? So I just apply this to this guy. Does this make sense? So the sort of the probability density for energies that have high fitness increase more than the ones that have low fitness, right? So and that's basically how it should change. Who is not lost? Okay.
Makes sense, but
All right, so we can go back to what we had for the frequencies, right? So ddt of x, when we were writing this down, right, is basically fitness minus the mean fitness. So x was the type A in that case, FA minus the mean fitness in a population, and mean fitness was FA times x plus FB times 1 minus x, right, times x. So that was what we had in single allele, right? So this is exactly the same thing. Now, if you sort of calculate this thing, this would give you delta FAB 1 minus x, right? And that was what we had. And so this is by sort of by analogy, the mean fitness minus the mean fitness times the probability density at that energy value.
Do you understand what we're doing? Like, do you understand the difference between the phenotype and the phenotype?
That I think that is fine. But so the main difference here, right? So we are sort of working everything out in a continuous regime. We don't have only one frequency value. We have a whole probability density, and we are integrating over all these phenotypes, right? So these are all sort of, you can think of it as approximation because your population is still pretty discrete, right? So there are n individuals and they have n numbers. So if you were to write this thing properly, you would write down the sum, which is somewhere up there, and how individual x's change, which they change like this, right? And then you can do the same approximation we did up there, and you produce this integral. So I'm sort of skipping that step, but we know how x alpha changes, right? That it changes like this, and so you can really build up as a sum and then go from that sum to the integral. Maybe this is more clear now? All right. Yeah?
Maybe elaborate on this probability distribution of the a little bit, because I feel like what we did on the top blackboard was to assume that it's Gaussian.
Yeah.
And then on the bottom blackboard, I don't see the connection to the time derivative.
So, okay, so where does WE come from? It's the same thing as x, right? Just in the continuous limit. X is the frequency of a type. Right, so the thing that is written in the bottom, you could actually write it down like the first line, right? E alpha x alpha, right? So this is the frequency of a type with energy E alpha, right? And so, E alpha is a constant. So you make a derivative of x alpha. We know how to do that. It's the fitness of alpha minus the mean fitness. So this would be E alpha. So fitness of alpha, that depends on E alpha, because fitness only depends now on energy, right? So fitness of E alpha minus the mean fitness, which we calculated up here, right? Times x alpha, right? That's what we have here. And you can go even one step further, right? So you can basically write this down. This is minus C0 E alpha minus E star square. What is this? This is minus C0 gamma minus E star square minus C0 delta, right? There's a x alpha, so you need to sum over all alphas, and there's a E alpha here. So this is a sum in principle, right? And you can now do a projection from this sum to an integral. a Gaussian integral just for simplicity to solve it. But you don't have to, right? I mean, in this case, if you assume it's a Gaussian, you actually really don't have to. Is this now good? So I'm going to sort of skip this step from now on, right? So it's sort of that's the rationale, right? Okay. All right. So if I put down what this is, like, which is exactly what we have here as a function of E, I make the integral. You can do it, it's a simple Gaussian integral. And what you end up having as a solution here, can anyone guess what the solution should be? We have a sort of scheme of how to write down solutions due to selection, right? There's a sort of covariance matrix always times the selection coefficient, right? That was what we had there. It was G alpha beta times some S beta. And S beta was a derivative of your mean fitness with respect to your X beta, right? So what is the analogous to the covariance matrix in the phenotype description? It's a phenotype variance, right? So that's the only variance you have in the whole setting. Before it was, the mean was a mean frequency, the variance was variance of frequency types. Here the mean is mean of phenotype, the variance would end up being, and we're going to show that that's the case, the variance would be the variance of phenotype. So the G alpha beta or G gamma gamma in this case, right, should be probably delta. And we can show that. And then what's the derivative? How do we do the derivative of fitness potential? So what's the fitness potential? It's a mean fitness, right? So the derivative of fitness potential with respect to gamma is minus 2C0 gamma minus E star. So basically, what we expect to get out of this would be minus 2C0 delta gamma minus E star. Right? If I put delta at the end or in the beginning times delta, this is your covariance matrix, and this is the derivative of your fitness potential, or it's a selection force.
So how do you see that?
Well, I mean, you just calculate that integral. Yeah, exactly. You get this. So this is the derivative of this mean fitness with respect to gamma, right? This term. This is mean fitness. Or that. So with respect to gamma, you get this term. And this is your covariance. Okay. But you can calculate this, right? So that's the easy thing to do. It's a Gaussian integral to calculate. All right. What's the state? Yeah.
You're basically saying that the rate at which the mean phenotype of population is moving is proportional to the variance
Yes. Of the phenotypes.
of the phenotype.
Which is due to selection, right? So selection selects from the variance. Before, the rate that your frequency was changing was proportional to x1 minus x, which was the variance of alleles in your population. Now the rate that the mean phenotype changes is proportional to the variance of phenotypes in your population.
Okay, maybe write down the final equation. I think then it will be easier for people to see. So now you have to your left to do this integral, and at the end of the day you get the change of
Yeah.
gamma.
We have a real.
Well, you have it everybody understands that you still have to do this integral, and you know how to do that integral because you're told how to do it on the top board. So
Is all right. Maybe we get rid of this part.
there's a third board, I
I think it's also full in the back. Maybe we write down the integral on that helps. It's also full. Yeah. So from yesterday. So all right. So I take the minus C0 out. So this would be basically E minus E star squared, which is f of E, minus the mean fitness, which is then gamma minus E star squared minus delta. Right. So, and then that's a W of del E, which is a Gaussian, we approximately assume is a Gaussian, D of E. No, C is out. Right. All right. So you can see. So okay. So you can see how it will.
and a square up there.
Yeah.
See, I'm following.
Okay. So the mean is gamma, right? So one thing you can do is you shift whatever is here with gamma forward and backward, and then E minus gamma squared gives you always the average of that gives you a delta, which is the variance, and then you get gamma minus E star square. So that's the easy thing to do, right? So this would be E minus gamma square plus gamma minus E star square minus 2 E minus gamma minus E star. So you sort of do all these tricks. I mean, this is, then you basically get the solutions. I think at this point, maybe you trust me that's the solution, because this is a Gaussian integral that one can solve. Just you move around terms. All right. Yeah? The question?
So the stuff in your integral is just you're trying to simplify the integral.
Yeah, I want to sort of move things around gamma, right? So that's what I wrote. So then we have the effect of mutations, right? So on gamma. And so let's say I want to understand how gamma changes due to mutations. That's an easier thing, right? So I can write gamma as sum over all types or all individuals in my population, right? The average of E. So N is the number of individuals in a population. Now alpha is just basically the identity of individuals, right? So that's a individual. And then you want to calculate the change in that due to mutation. So what we know is that mutations are random, and they do the flips from 0 to 1 or from 1 to 0. If your sigma is equal to 1, you do a flip. Your delta sigma would be minus 1, right? Mutation. Your delta sigma, so 1 to 0, would be minus 1. And if it is 0, from 0 to 1, delta sigma would be equal to 1, right? And so in general, you can basically write down your your delta sigma would be sigma minus 1/2 times 2, right, minus, right?
One of the three is 0, the top, to minus 1?
2, 0 is 0 and 1.
0 minus 1 for one of them.
So if you're 1, you get minus 1. If you're 0, you get 1, right? And so if I now put this up here, right, so my delta gamma would be 1 over n, sum over EI. And then they happen with a rate, right? So mutation rate mu. So you multiply it with a rate minus 2 mu 1/2. Right? So I take out minus 2 mu. Sigma I alpha. So EI sigma I alpha 1 over n, and then minus 1 over n 2n sum over I and alpha 1, right?
What's the sum over?
Sum over all the positions and the individuals, right? Sorry, EI. Right, so this sum basically cancels out the end when you sum over alpha, which are all the individuals, because there's no alpha term here. So basically that gives you sum over EI, and this is just our gamma that we start with, right? So this is basically minus 2 mu gamma minus sum of EI over 2. Right, and this is a thing that would have been very easy to guess as well, right? If there was no selection, if you only have mutations, the mean phenotype basically approaches the random state, and the random state here, if all the size were equal, so if all the EIs were 1, the random state would be L over 2. So in this case, because you have these quench variables EI, so it's a sum over EI over 2, right? Okay, so we have the deterministic parts figured out, right? So we have the selection, we have the mutations, and I sort of told you that G gamma gamma is equal to delta, and we're gonna write this down next time how it is equal to delta. But if you trust me for the moment that it is, then you can actually write down the full equation for changes of the gamma, right? So gamma 0 is the sum of EI over 2. That's how I defined it. So this is what we have. Okay, so I sort of rescale things, and I'm going to sort of discuss that, what these rescalings are. So C0 has dimensions, right? Because fitness needs to be a dimensionless quantity. So if you have a fitness landscape that looks like C0 E square, the dimensionality of C0 would be 1 over dimensionality of your trait. Now, if you want to sort of move into dimensionless quantities, which is good for a lot of different things, you can rescale C0 with a sort of factor that depends on, that scales with E square. And let's call that E0 square for the moment. So that's what the difference between C and C0 is. So C basically is equal to C0 E0 square. And then you do the thing here. All right. So the first term is the mutation on the left board. The middle term is the selection here. And the last term is basically the genetic drift. So your G gamma gamma, the fluctuations are proportional to the variance. Delta, and you have a 1 over n because it's a genetic drift. So it's like what you had, x1 minus x divided by n, right? So right now how I wrote it down is I said, okay, let's use a noise with mean 0 and variance 1, and then take the variance term outside. So that's standard deviation, basically. And there was an easy way of guessing what this standard deviation term should be. Does anyone have a guess? So what does it represent, right? So this is a sort of difference between ensembles of populations, right? So there's a noise at the level of ensembles of population. So it's basically saying if I'm trying to evolve gamma, what the fluctuation of mean phenotype should be if I do it many times, right? So across ensembles of sort of trials, what is the fluctuation of mean phenotype when I select from a population with a phenotype E? And I think you know from your first lab course, that should be just standard deviation of the mean, right? So fluctuation of the mean should be proportional to the variance divided by the number of samples you take, which is in this case n, right? That's where delta over n comes from. And we're gonna write down a similar thing for delta, not into details, but sort of trying to guess terms the way we started guessing terms here. But we have the tools to sort of write things down rigorously, and that's the point, right? So if things are not as trivial anymore, we can actually do it, because that's basically the machinery, how it should work. But the trick is, the other thing that you should sort of see from this is the evolution of gamma depends on delta, right? So evolution of mean depends on the variance. Evolution of variance will depend on the fourth moment. And if you're not, it doesn't close. If it's Gaussian, it closes, right? You close it at second moment, and the fourth moment would be proportional to the variance square. But in general, it's an infinite rule, right? So infinite moment expansion, and that's, we're familiar with that from physics as well. The other unique thing about this is that fluctuation of the mean and variance, and we're going to see that next time, happen on very different time scales. So variance fluctuations are much faster than the mean. And that's why you can do a trick of separation of time scales here, and sort of look at, treat variance as a fast variable, and just look at marginal distributions of gamma. Because in principle, gamma and delta live in the same space. And you have to look at the joint distribution of gamma and delta, and we can write it down. And there is, unsurprisingly, there's a curl, so there's no equilibrium in that space. But once you marginalize, then you obviously have equilibrium. And the solution of this equilibrium is, if you treat delta as a constant, which would be sort of ensemble averaged delta, as it is a fast variable, then this is what you know as Austen-Uhlenbeck process, right? You have basically a gamma that lives in a potential that has some its value in neutrality is around gamma 0, and there's a shift in the place that you put your potential, and you shift it closer to E star as your selection increases. And the solution will become some Gaussian form, but you also get the same Boltzmann relationship, because there's a delta and delta, so it's a potential. You get the same form as we got from Kimura, relating distribution on the selection to distribution in neutrality, with the same n coming up as inverse of your temperature and mean fitness as your fitness potential.
I think we should stop.
Yeah, I think that's the time to stop.